Hey everyone. Hey Martin, welcome.
Hi.
Let's wait for just 1, 2 more minutes so everyone is
Sure.
there. How do you feel, Martin? Look a little bit exhausted. I know.
No, that was last week.
Oh yeah, I know you
Last
had a
week I had
crazy
three
week.
conferences. That was like a good amount. That was too many. Two is okay.
Oh, two is fine. Okay, I'm letting more people in. So yeah, we will have some people here in Zoom. And we will also probably have some people on YouTube. So there will be chance to ask questions after Martin's talk. So let's not ask question during the talk, and let's have like a good discussion after, because the the questions are very hard and very important. So I will also monitor YouTube, Martin, so you don't need to do anything. If someone asks
Oh,
something there, I will be the person who will ask it. Okay, so let's welcome Martin. He's a CSO and founder of Gordian, and also founder and president of Norn Group. I hope I remember everything correctly, Martin. If I'm not, you will please correct me. Um, Gordian is one of the leading longevity biotech companies, and Norn Group is a non-profit which is accelerating longevity and doing a lot of useful work for the field. Today's talk will be focused on Norn Group and on the new big picture, which is kind of a roadmap. But yeah, Martin will
getting
talk
there.
more about it. Okay, Martin, floor is yours. Welcome.
Yeah, sounds good. Thanks, Alexei. So, what is Gordian? What is Norn? I'll talk a little bit about that in the presentation. Gordian's a biotech company. We are finding an answer for every disease by using pooled in vivo screening to systematically map out, create an atlas of basically all druggable targets in each disease area and which ones are best, and then develop drugs based on that. So I think that's important because there are many ways, and we'll get into this, there are many ways of making a drug better, including with AI. There are not many ways of finding out what should be done. And so we kind of have to fill a niche hole there. And then Norn Group is the idea is that there should be a plan. And let me start sharing here. Do a full screen. All right, cool. You're seeing nothing. Is that correct? Good.
Yeah,
You're seeing some.
we see some trees.
We're just going to look at trees the whole time because stress is bad. And if you're not stressed, you'll live longer. No. So we're going to go back to 2010. And so in 2010, I was just coming from Denmark, where I was born and did my undergrad, to start my PhD at the National Institute on Aging in Baltimore. There's a Bayview campus for probably not even my phone back then. It was probably like a camera that I physically carried around. Um, and I went there because I had already, for maybe 5 years at that time, been thinking this is the most important problem. It's the disease that we all have. It's the biggest driver of top 8 causes of death in the United States. And that's, setting aside all the morbidity and all the pain that's coming. So we need to solve aging. Don't need to tell everyone here that. I assumed that there would be a plan somewhere. Someone would have a plan for this. And who else but the National Institute on Aging of the United States, which is producing most of the research on aging. They must have a plan. I will humbly contribute to the plan, and we will get everything done. Unfortunately, I showed up and there was no plan. There was not even necessarily a desire to solve aging. There were a bunch of labs working on interesting stuff, but yeah, you've probably all had this experience in your lives. So this is stressful to me because I like having a plan. I also play board games sometimes when I'm not doing a for-profit or a nonprofit. And if I have a plan, I usually win. And if I have no plan, I rarely win. And the same is true, if you're doing sports, whatever it is, you don't just show up to the field and then let people do stuff. You should have a plan so that you know what is important to do. So let's make sure that there is a plan for the longevity field. That's basically the reason that Norn exists. I want to make sure that someone is looking at the big picture of how are we going to get healthy years of life? What will it take to get there? And make sure nothing falls through the cracks. We don't accidentally solve half the problem. So we're going to talk about that picture. What would the plan look like at a fairly high level, of course? But what are the key things in that plan for roughly half an hour, and then we'll have a lot of time to talk about it. Our plan's going to involve defining what we actually want to accomplish. Very important part of a plan is that it defines the victory condition. And so for me, I'm proposing that the number of healthy years that we add annually, so the amount that we extend healthy life expectancy per year, is a good metric. You could also go for quality adjusted life years per dollar. That would be reasonable insofar as it brings in sort of a cost aspect. However, you could get a whole lot of those without extending lifespan by just removing all the unhealthy stuff that we're doing. So I like this one. Then we should try to understand the production function for this output. How are we getting those? Then, because everyone here is trying to do an AI hackathon, we should maybe look at where AI has an impact or could have an impact, to make it topical. Oops. And in order to increase the output of the production function, we have to find ways to make the input for this output abundant, right? And the input is basically ideas. It's something that we could do that might give us more healthy years if applied to a human being. And then we have to figure out what are the bottlenecks in this production function. What are we currently limited by, and what could we be limited by if we make some changes? So we're going to go through all that fairly fast. Where are we right now? If we look at the output metric that I defined, healthy years of life, we've had, something like 200 years of just steady increase in the number of healthy years of life that we are getting. And this is from Our World in Data, great source. Each of these lines, these horizontal lines, is someone's guess at where we're going to peak out. And you can see that they are wrong always. And sometimes they are hilariously wrong in the sense, here, this is going to be the peak. Already, the reality was higher than the prediction when it was made. So it's possible that we will peak out at some point. But so far, biomedicine has, through innovation, been giving us roughly 0.1 years of healthy life per year. So that's nice, but it would be nicer if the number was above 1, because then we would be solving problems faster than we encounter them, on average at least. So how do we get there? And where are we at right now from the longevity field? All of this progress is coming from biomedicine broadly, not necessarily the longevity field. The way this works, some of you are familiar, some are not. You have a research stage where we are generating ideas for new medicines. And so a lot of this is in academic labs, not all of it. And we might come up with, hey, senescent cells, they seem to be bad. Maybe we should clear them. That's one idea. It could be IL-11 seems to be important for a bunch of age-related diseases. Maybe we should make an antibody for that. That's another idea, so on and so forth. All of these are ideas. You can even generate data, make a genetic mouse, find something in human populations that suggests that this is a real idea, but it's still just an idea. It is not a medicine that we can sort of apply to a human being. In order to get there, we have to go to the drug development stage, which is typically biotech, sometimes pharma, where we are instantiating these ideas into something physical that could be applied to human beings. And so the traditional version of that is a pill full of a specific chemical that will go into your body and bind to certain proteins and inhibit them and so forth. But it could be other things as well. It could even be some weird ultrasound thing to clear plaque in your brain. Either way, this has to happen first. Then we have to go into humans and show that what we thought would happen actually happens. We're pretty bad at that. Usually it doesn't. But therefore, in fact, this is a necessary step. So this is our sort of production machine for healthy years of life. And if we look at the longevity field specifically, this is of course a bit subjective, but anyone who has sort of a label of longevity, anyone who's at or funded mainly by the National Institute on Aging, will lump all those things together. At Norn, we've sort of cataloged how many ideas are there per year. We should put this out somewhere, but basically just count up everything you can think of, divide by the number of years, you're at some single digit number. So we're only having a few ideas per year that could be a therapy. And you can go through that in your head while we're talking and say like, well, how many can I name from last year? Then of those, I would say quite a high fraction. All these numbers are rough estimates. Don't do math with them, but use them to understand the field. Quite a high fraction of those ideas are actually getting turned into a company. It's sort of hard to find anything decent in the longevity field right now that's not being commercialized in some way. And I think part of that is because this number is relatively low and there's some interest now. The timeline for those companies to go from idea to instantiated product is something like 5 to 10 years. We can look at Unity. They started in 2011 and they started going into trials in 2018, I think. You could look at partial reprogramming. The first major paper was in 2016 and New Limit is not yet in trials, but might be within a couple of years. So that's on the order of a decade, right? And then what is the success rate? So this is a big deal because right now we don't have a success coming from the longevity field. So we don't know what the success rate is. So I'm going to put it somewhere between 0% and then 1%, which is the overall success rate for medicines in general, right? So just biomedicine going all the way from preclinical research ideas through clinical trials, it's something like 1%. So we're somewhere there. And then let's just assume that a successful application of idea, clearing senescent cells, reprogramming the cells in your liver, might give you 2 years of healthy life. I think that's a pretty big number. Most things don't give you that much for the average people, but on the other hand, mice are gaining quite a lot. If these numbers are correct, and then you'll do 10 years of trial, then the longevity field currently is contributing somewhere between 0 and 0.04 years per year. So on the bright side, on the bad side, that's not much, right? We're far from 1. It's not super impressive. On the bright side, it's not that low compared to what all of the rest of biomedicine is doing. Now, we don't actually know what the number is. We're just estimating that it's somewhere in this range. We'll note that there's a 20-year lag before ideas become reality. And really, most of the ideas in the longevity field started popping up from the 2000s. So we're only getting to the stage where some of these, have gone into trials and might be successful. So that's where we are right now. So if you're like, oh, we're done, don't worry, longevity escape velocity, bad news for you. We have to act in order to make longevity a reality. Good news, in a sense, and bad news, is that biomedical research in the US is something like $250 billion per year across all government funding, all companies, and all nonprofits. Globally, it's more by 50%. Aging biology is less than 1% of that. So if you count up, the NIH gives 400 million per year, something like that. And then there's ARPA-H and a couple others, some tens of millions there. All of Europe is 100 million, 150 million maybe. There's a few things in Singapore, Japan, Middle East. And then there's some companies, but most of the companies, I mean, it's not that much money, right? Like there's like a one or two double-digit million raises per year, and then Altos and Calico have something like $100 million budget per year. So somewhere in between here is what we're doing. Very small. And so if we're actually having like contributing 10% while only getting funded this much, that's pretty cool. So like that's something to hold on to. Our field has promise if we can make things work, if we can make the success rate not zero. It also means that like if you do something that increases the size of some field, this could be quite significant. The number of labs might be on the order of 500. This is very rough. It might be 1,000. I haven't found a great way to quantify it. One ways you could look at it is like how many grants are awarded by the NIA and other institutes in Europe and so forth. Some of those might go to the same lab. Some of them might not quite be longevity. Some people might get grants from the Cancer Institute, but they're really longevity. But it's not that many. And then maybe each of these labs has a few people in it. Some of them are big. George Church Lab, obviously very big, although not mostly focused on aging. Some have like 2 grad students. There's 5 people in each. You know, if we have a couple thousand people in the aging field, and then if we assume that, you know, there's a range of talent in the field, some people are not very good. Some people are maybe quite good. Few people are very, very good. Most of the very, very good people are doing AI for CRISPR or something else that pays better. But that means that if a large fraction of the output is coming from a small number of the people, if you just add one person, if you believe in like a 10x person, then you might be improving the field by 1% just by adding one person. You know, if you don't in biology, then maybe it's a smaller number, but it's like a meaningful number. And so some of the stuff we've done at Norton Group, my nonprofit, has simply been to try to increase the size of the field. There's a couple of different programs that we've done. It started honestly with what's called longevity apprenticeship, which was just 5 people, sort of like grad students, but not for doing science, for doing meaningful initiatives in the aging field that I mentored. That resulted in 3 companies that I'm aware of. Impetus Grants was run by Lada, who was in this program for several years. So I think that was very successful, but not super scalable. We've converted it now into what we're calling Norton Nexus, which is a bigger community that are actively engaged in making progress on longevity. We also launched a Talent Bridge program, which helps talented individuals get to labs in the US. We've done 2 awards so far. I'm going to continue to do those. Might be relevant for some people. You can check it out on our website. And then, of course, we did Impetus Grants, which was sort of fast grants for aging, specifically focused on low bureaucracy, high potential impact research. Our core metric is whether there will be conference sessions based on research that we funded first in the future. This is a very lagging metric. Leading metrics that you can look at, you know, papers, all this kind of stuff, look pretty good and distinguishable from NIH funding. And also, I'm proud that something like 20% of the labs that we gave grants to were not previously working on aging, but seem to be continuing to work on aging now. Now, if this number is correct, you're adding like, again, several percent to the size of the field, but just by providing these bridging grants. So I think here's one action area that just in general should be motivating to people, that like we can make stuff happen. Of course, we should make good stuff happen. So that's more challenging. Now let's talk about why is this true, right? Why is there so little money in longevity? Should be a lot, right? And if you sort of read journalists, whatever, like then it seems like there's a lot, all the billionaires, whatever, but it's like not actually a lot. It's just like dramatically outweighed compared to, it's sort of like death by terrorist, right? Doesn't actually happen hardly ever, but gets a lot of attention. So you think it's a lot. It's really not very much at all. Why is that? Well, in general, for investors, financing follows faith in rewards, right? So if you look at these 3 circles, they represent respectively 10-year US Treasury bonds, which take 10 years to mature, biomedicine in general, so running through clinical trials and all that stuff, which is also on the order of 10 years, and then longevity R&D, just all of it, even the preclinical stuff, right? Small, large, $5 trillion per year. Incidentally, also what we spend on healthcare in the US per year, roughly, versus $250 billion per year. So 20-fold less on research than on paying for diseases or putting money into the treasury bonds, and then less than 1% of that. Why is this? They all take roughly the same amount of time, but the probability of getting a reward out of your investment is very different, right? If you put money into US Treasury bonds, you're almost guaranteed that you will get a return on your investment. It might be lower, maybe single-digit percents, but you know that it will happen. So you can plan around that, and you can, whatever, if you have $100 million, you can live off, $4 million a year by having them in treasury bonds. It's going to be steady. Retirement funds can use this, right? If you invest in biotech, it's way riskier. Success rates, depending on disease area, 5 to 10%. So that's far riskier, but you have some sense of the success rate. And you can do math on what is the market for this indication and like what would it mean if we succeed. And the return on investment is something you can estimate at least. For longevity, nothing has succeeded. So how hard is it? The probability of success is a total question mark, right? And I think when it is a question mark, not only is it hard to model what your ROI might be, but also it could be zero. We could be like just very far from anything working. And I think this is a large part of what deters investment in something that otherwise should be hugely important, right? It's driving most of this and this. So if we could change it, then that would have a very high societal ROI, and I'm sure a very high financial ROI. We just have to believe that a success is actually possible. As long as we have no feedback, then the investment into this field has to come from people who are sort of mission-oriented, have disposable income. We should do a little bit here just because it seems nice, but it's not really something where a cynical capitalist is going to go put money in. We should change that. This is one of the key numbers. If we change this number, then it can become something that is like mainstream rewarding. So to summarize that, we have our whole ecosystem where we do research, we do drug development, and then we go into human testing. If we can demonstrate, put a number to our probability of success from clinical trials, maybe even from the drug dev stage, and we'll get back to this later, then you will get more funding and you will get more people who work on longevity because it'll no longer be sort of like a whatever dead end for weirdos, right? Which is not totally unfair, but maybe a little unfair perception that many people have of the longevity field. And then because the field is so small, that could lead to several-fold increase in the number of ideas we have. And so now our production function is happy. It is being fed more abundantly, and we could get more years of healthy life out. There's a downside here to taking this strategy to improving the size of the field. We'll get to that in a minute. But first, let's look at how could we do this? How could we get our first probability of success? So obviously there are companies. There's maybe, I forget what Carl's list is up to, but at least 100. Some are more serious than others. And so as those go into clinical trials, and some are in clinical trials, some are close, we could just get some successes. Some of those might be branded longevity, or at least turn out to be longevity, similar to how GLP-1s turned out to be effective multimorbidity drugs, and perhaps longevity drugs. And so that's one way we can do it. It is quite expensive and takes quite a while, and it's sort of dependent on what's being fed in. We can look at some potential shortcuts here. Probably most of you are aware of Loyal, and there are some other initiatives as well that are trying to go into pets instead of humans, because you can run the trials faster and cheaper, and you can demonstrate a real lifespan outcome in a number of years. It might still take a decade. It might take less for them to get enough signal, right? They're hoping to make dogs that live 8-ish years, live maybe 1 year longer. So it's not going to be a super dramatic effect, but with enough dogs, they have 1,000, I think, enrolled in their trial. This could be a readout. So that would be cool. If that happens, we might get a lot more investment into the field. Another thing is you can find clever ways of doing the human studies. One of the grants that we awarded through Impetus grants is for a clinical trial that's testing rapamycin for reducing decline in ovarian aging. And so the idea here is that women between, I forget their age range, I think it's 38 through 45, that are still menstruating, they're going to go into menopause. You're going to be able to measure ovarian reserve, AMH and FSH, different hormones. And you could potentially see a difference in women treated with rapamycin if indeed rapamycin is an effective longevity drug that also affects fertility. And this is a trial you could do fairly fast. This is going to be, I think, 1.5 year trial, something like that. I think it's going to be done end of this year, if I remember correctly, fairly soon. And if that succeeds, then you have several things going for you. One, well, all the people who undergo menopause might have something that they could take. They'll need to run additional trials before it's like this is something people start prescribing, but the proof of concept will be there. But also, this could give us a way to run an aging trial without having to wait a human lifetime, right? So you can imagine this or other schemes that allow aging trials to be run faster. And then we could because most trials still fail, we want multiple shots on goals to get to this first success. So I think this is an area where you can have sort of a very high impact by doing, literally anything. There's not that many things that are happening. But the downside of this approach to enlarging the field is that if you do something now, and then you produce more ideas, and then they have to go through the timeline, you're still looking at like 20 years, maybe 15, before you have something cool that, you can give to your grandmother. And so I think this is an important feedback loop that we should cultivate in the strategy to increase the output of the longevity field. But we should also consider what else can we do that might give us an impact sooner. Okay, so I'm going to just pause and go back, summarize the numbers that we've looked at so far. The first one, which is our output function, is years of healthy life added annually. It's at 0.1 now, or it has been historically for the last 200 years, very consistently. The aging field might start adding to that. You could envision that makes the number go up a little bit. You could also envision that, no, that's just the next iteration of finding new ideas and the number is going to stay the same. If we want to increase that number, the key other numbers that we should look at are, what is the hypothesis output rate? How many new ideas are we having per year? How can we change that number? And how good are we at selecting ideas that work in humans? We'll get back into that in more detail. But basically, if we can pick winners, then we can demonstrate that this is a real thing, and then we can get a lot more funding. And there is a cycle time overall that's kind of a hassle, right? Fairly long and very annoying for anyone who has a mission of doing stuff by, let's say, 2050. You really have to get going now. 2060 gives you a bit more leeway. So those are the numbers that I think define progress in the longevity field. If we look at it as big picture, a field, a production function, an Andy Grove breakfast factory, rather than focusing just on the individual scientific bits and pieces. Today, the number of ideas is fairly low. The probability of success is a question mark, and the lag is 20 years. So let's get into what are the areas where maybe AI can make a difference here. Now I'm getting into stuff where maybe you have some idea that I haven't had. And so not meant to be an exhaustive list, but a view from someone who engages in aging biology and in drug development and thinks about the big picture on where we can actually make a big difference in the short run. So this is still our production function, but what if intelligence is really cheap? What if, Demis and Dario are right, and we will have infinite intelligence very soon? Will that immediately solve disease in 10 years as they are dreaming of? Default answer, no, sorry. And part of it is, what is a disease? And so what we want to do is we have this disease state, let's say, and we want to go to a healthy state. So we want to take a human body from a diseased or old state, and we want to go to a different state. The problem with that is that a lot of our data looks like this. We have some brains from some Alzheimer's patients that died, and we can see maybe what they look like at the transcriptome, at the proteome, whatever, but like just a snapshot. And we can see this over here. But biology is not just some like linear addition of things. There's a lot of nonlinearity in biological systems. That's how we, prevent ourselves from turning into a giant blob of teratomas and so forth, right? Everything is tightly controlled. Everything has feedback loops. You get sick, and then you get a fever, and then the fever turns back off. You get proliferation of immune cells. They go in, they kill cells, but then they turn off. You regenerate. Everything has to like come back to the homeostatic starting point, right? That means that it's really hard to map out what's the path from A to B. And I think if we want to utilize cheap intelligence to help us say, what should we do? What is the idea for, curing a disease or bringing a human to a younger state? We need to start poking the state. We don't want just the snapshot state. We want like, what if we do these different things to the state, what happens? And then we can use that to map out what are the paths whereby a disease state can get to a healthy state. So we need a lot of data of that sort. That is what we are doing at Gordian. Some others are doing as well, although not as many as I would want. What we're doing at Gordian is to use what we call mosaic screening, which is taking a library of different perturbations that up or down regulate genes. They're all barcoded. They're in AAV viruses. We put those into living animals that have over time progressed to an AIDS-diseased state at a subsaturating dose. So you get these individual cells within the organ that have been perturbed in some way and effectively serve as independent cellular experiments, just like when you're doing high throughput screening in plastic dishes. But instead of the experiments being separated by something completely unnatural and that takes away most of the interactions that happen in the body and that lead to fibrosis and immune dysfunction and so forth, the separators are the actual disease tissue that comes complete with all those interactions. Importantly, even if we don't know what those interactions are, even if we don't know how aging works, which was kind of the capitulation that after, like 15 years of working on aging, I was like, man, I still don't know how it works, but I have discovered that nobody else does either. This is a Gordian knot, and we need to find a way to make progress without having complete understanding. So that's the name of the company, and thus focusing on making this platform possible, where we do this, we pull the cells out, and then we do single cell transcriptomics to read the state of the cells given all the perturbations. And so basically, we are mapping out, we're poking with all these arrows, and we are mapping out what is the transition. We're going sort of disease by disease initially, and then we'll transition into old versus young when that's a sort of feasible de-risked business model. And hopefully we'll generate the data that then with abundant intelligence can start to make suggestions around how do we go from A to B? How do we cure this disease? So that's something we can do. What about at the sort of drug development biotech stage? Typically, what is a longevity biotech? It's a company that takes some idea and then does this instantiation into physical molecules, and then figures out, well, what are we actually doing here? What clinical trial do we want to run? And builds confidence that they're going to get good stuff out. We can look at NewLimit as an example. Their idea is partial reprogramming, that we can use some combination of transcription factors to turn old cells younger. They are delivering those transcription factors as mRNA combinations because they want more than one in LNPs. They are using AI to optimize what combinations and doing a lot of learning there. Then they pick some disease. The first one they've picked is alcoholic steatohepatitis. So basically your liver getting fibrotic and wrecked from drinking. And then they have a bunch of mice that they give alcohol, and they show that the mice are better off after they have this treatment. They're trying to do other diseases as well. So this is what every company does. And like, you might say, oh, this is not very ambitious. You're just taking one idea and you're going to cure some like drinking shit. What? This is lame. There's a good reason why that is what they are doing. All this stuff is really hard. And most companies are like, oh, I'm just going to go do this. And like, we're going to go to Honduras and we're just going to make our own gene therapy. It's really easy. I read a paper once. Those are all going to fail because biology is very unpredictable. And sorry, I probably insulted someone on the feed. Maybe they won't, but probably they will, right? Like, we're not just like spending a lot of money on biotech because we're stupid and we like burning cash, right? It's because it's actually hard to engineer biology. And so that's where New Limit are like, well, we're going to use large data sets, large scale perturbations, and predictive learning to do this much better. And we're going to be a platform and it's going to be cool. And this is just the first thing. We're going to do more later. And so they're doing this. I stole this from one of Jacob's blogs, active learning that apparently is able to give them better combinations of transcription factors for doing this thing. So they're able to make this work better. Their AI is able to accelerate this step, making molecules. And that's generally true. If you think of AlphaFold, if you're making a protein drug, you can simulate, protein folding, protein interactions very well now. Antibodies are coming online as well. For small molecule drugs, chemicals, you also can use the structure generated with AlphaFold to sort of predict binding. So there are a few places where we've gotten more efficient at actually making the molecules by using AI. We're also sort of making them from scratch, Gen AI molecules or whatever. I think most of what I've seen for that is basically like, well, if you already know that this works, then you can make something that also works. And you will be able to circumvent the first patent, the patent of the first people. Less so like we have no idea what will work, but AI has generated a molecule that just works. Like this part of it, or even the idea part of it, we haven't really done that yet, to my knowledge. And it makes sense because what is the training data? What is a disease? If we do a brief detour here. Oh, no, wait. Sorry, I hit those slides. Here, hold on. I'm going to unhide those slides. So let's get back to why this is hard to use AI for. It's because where does a disease live? Biology is made out of a bunch of intracellular components, form proteins, form cells, form tissues, form organs. Can you point me to the disease? Where is heart failure? Is it in here? Is it this one? But it's really like the beating of the heart, right? So maybe it's like the whole thing, because the ultimate disease that we are trying to cure is like, what is the ability of the heart to pump blood out, right? But there are changes at the cell level. Oops. So you have all these changes at the transcriptome, DNA level. Some genetic diseases are just here, but complex diseases are not, and aging is not. And then there's all these differences at the cell level. And then, yeah, the tissue changes. Where is the disease? Can we point to it, right? And therefore, if we like struggle with that, how do we tell the AI that like this is the thing you're trying to solve? We can put a label on it. We can say Alzheimer's disease, but that doesn't actually mean anything, right? Like that's just us putting a label on something. It might not even be one thing. We can put a label on something and call it cancer, but there is not one cancer. There are many, and they have different, metabolic rates and ways that we can treat them. So I think that's the challenge that makes it hard for AI to sort of generate new ideas and just say, here's what you should do. And that also makes it hard for AI to predict what's going to happen in the trials. We don't have the training data for those things. Here, I don't have a solution for you, other than we should focus on what is the data that we would need. How long will it take to generate it? If it takes us 10 years to generate the right cohort data, maybe we should start now. But there is one area where I am pretty confident that we should generate data, and that is like how well, when we do stuff at the animal level, how well can we predict that? Because if we are good at that, if we have sort of good, like evals for saying what will happen at the physiological level, then we can generate more faith even before clinical trials. And so here we can get to clocks, right? Aging clocks, everyone's familiar. Something that measures something molecular and then tells you your biological age, which we wish was something that told us how long we were going to live, but we're not sure if that's actually what it means. The problem is that most clocks currently are doing DNA methylation or whatever else at some time point, and then they're checking, hey, how well does this predict the age of this individual? Some of them go beyond this. They predict sort of time until morbidity, but all with non-interventional cohorts. So none of the clocks have been validated in a way where you say, hey, I made this change to the biology, and now the clock has changed, and I'm going to see whether that means that you actually live longer. Because that's not necessarily true. If we use a wristwatch to assess time, it can be really good at this thing. Like if I look at my watch and like 5 hours from now, I look at it again, it'll always say 5 hours later, right? It's well built. It's probably European or Asian. It's not American. And it's really good at predicting these things. But what happens if I just like wind it back 5 hours? Did I do time travel? So we can have something that is very accurate at predicting things in sort of like a steady state, but that is not good at responding appropriately to interventions. The equivalent here would be that you have your methylation pattern. For many of them clocks, it's a black box, right? So we don't know what is actually being measured. And then you give some drug, maybe you give a, demethylase, and then the clock goes down. Is the person younger? Or did you just change the surrogate that you are measuring with the clock, right? We need to be able to assess that. And in order to assess that, we need to do an experiment of this sort where we take some animal and we do an intervention and a control, and then we measure whatever our clocks want to measure. And then we look at the actual endpoint that we care about. This could be incidence of disease or lifespan or whatever. And we see across many interventions, how accurate is this clock? So this would give us something along the lines of like a CASP for, biological clocks. How are we doing, right? Are we actually getting better and better? Or are we just like making 100 clocks, putting them all in a clock shop, putting some of them for sale, never going outside to see if they line up with the sun, right? So we don't actually know what's going on, but we've got a lot of clocks. This is something you could do. You could do a design where you basically just like take interventions from the NIA-ITP. And so that rapamycin is going to extend lifespan, and resveratrol is not, and so forth. We have some that work. We have many that don't work. You can generate data for all of those and then take blood, do omics. You can do methylation because everyone's doing that. You could do proteomics, which may translate better to humans. And then you can put out a training data set, hold back a test data set. Whoever has a clock that they think is good can say, hey, I think I can predict the lifespan of these animals without knowing what the intervention was. I think I can predict the lifespan. That's what we want, right? We don't just want to like track your chronological age. We want to be able to predict interventions if we want to use these for like longevity drug development. And then you can score them with a test data set. And then you can put that out on a public website. So now you have your CASP or your consumer reports for how we measure biological age, at least in mice. It would be nicer to have it in humans, but in humans, we don't have the interventions to say. And you can do this off 24 different interventions so that you can actually say, do you respond correctly half the time? Maybe there's some interventions that affect the clock that, inappropriately. Or maybe you can only predict caloric restriction, because whatever, the training data that you used in humans was like basically confounded by diet or something. And then we can see how well we're doing. Maybe we'll get bad news and it'll say, maybe we'll get good news and they'll say like, oh, here's this clock, it's pretty good. You guys can stop doing lifespan experiments. Maybe we'll get bad news and it's like, they're all kind of bad. We need to do better. But the bad news is better than the current status quo, right? Where we basically have to assume, I think serious people don't replace their lifespan experiments with some clock because we know they have not been validated, right? Like internally, at least, we know that. You can do all of this for a couple $1 million. That is not a huge amount, right? So this is something that we could do and that could give the field solid benchmarks. Again, what can we do right now? If we then have those clocks, now we can cut the lag. and we can get better at picking good ideas. We can then expand it and say, oh, actually, we want to score all these for how human relevant they are. Maybe the proteomic clocks are better because they're not picking like random methylation sites unaffiliated with genes that we don't know what they do. The proteins we can map from mouse to human. Bonus points there. So that's something we could do. And that's something where if we generate this kind of data, now we can envision that intelligence can actually help us, like do better, come up with new ideas. If we have some interpretable clocks and we have the data on those interventions, let's just apply intelligence. So where we are now and where we could go, I think no matter what, we have to increase the rate of idea generation somehow. And the sort of default assumption that like just because AGI that will happen, I would not bet on. I'm paranoid, so I would not bet on that one. There are ways we could get there if we have enough automated labs. We could get there eventually. But I think also just like think about what is the data that if we generate that over the next 2 years would be maximally useful for drawing conclusions around disease. We should be thinking about like what are the layers of abstraction where we want to generate paired data sets. If we do a whole bunch of single cell transcriptomics, like we're doing and like many are doing, and we're making virtual cells out of that or whatever, is that enough to speak to disease, or do we need to pair that with like tissue level characterization of function? I don't know what the right answer is for how many layers can you span. Is it enough to like build a virtual cell? I doubt it. Is it enough to have a virtual cell coupled with like heart function and so forth for a lot of different instances? Maybe. We should be thinking about that, and then we should generate the data proactively. Worst case, we have some really nice data, right? Best case, we've skipped ahead by a number of years. So that's something we should do. We should make the number of ideas 100-fold higher, which is definitely not at all easy, but something that could happen. Gordian's already screening and generating in vivo efficacy data. I think I haven't been able to like find the secret numbers, but I'm pretty sure that in a given indication we work in, we're doing more in vivo efficacy tests than like all pharmas and biotechs combined. Then, if we generate lots of ideas, our new bottleneck becomes, okay, how many of them do we want to fund? How many of them do we want to put into trials? Which of these ideas are good, basically? And I think that's where we should focus on, like, how do we determine whether an idea is good before we go into humans? We need to build aging biomarkers that are human compatible and that we know can like predict stuff here. We should also be thinking about which animal models are relevant if we want to use animal models, right? What are the most relevant data sets for predicting which ideas are good for longevity? Because if we have many ideas and we're able to tell which ones are good, then I think we will be okay. And then we will move somewhat faster because we can make molecules faster. It may also be that if we validate biomarkers for FDA use, we can use those in the trials, and then we can dramatically speed up the trials. That is a long process. Like that typically takes also, like as long as it does to develop a drug, and it's kind of a commons good thing. That's the kind of stuff that Norn is focused on. The starting point is to generate the data that this is like plausibly a biomarker. And so I think the sort of clock assessment, clock benchmarking program can be a good start. And then combined with, figuring out what are the right human cohorts to generate data from. Do those already exist? Can we find that and then use that to show the FDA and validate a surrogate endpoint? It's important here that like, if you haven't gone here, if you haven't done this, or at least read heavily up on it, like, you will probably run into like, oh, that's the process. I should have done something differently 5 years ago. And so it's important that we have like stakeholders at all the different levels, and that we talk to those people. We talk to people who have actually gone through biomarker qualification and know like how do we do this right, so that we can do this, generate data over 10 years, and then actually make use of it 10 years from now. So let's say that we get 100x the number of ideas. Maybe we're only trying 20% of them now instead of 50%. Maybe the lag is now at the lower end because of AI or China making the drug development part much faster. Let's say we get to the success rate that just biomedicine in general has. So we're not doing better than like cardiovascular or whatever, but we're doing as well. keep the impact per drug, and then reduce the trial length a little bit because we have better biomarkers to 7 years. Now you've got a 12-year lag, and every 12 years, you would be producing something on the order of 0.8 years per year. So that's pretty good. Maybe I want double that, but like now I'm starting to feel, less worried if we could get there. And that's just by doing a few select things, right? Like this thing is already happening. Gordian, please like help us come up with some better additional ways to like generate more ideas and then focus on how do we validate ideas. Then we could plausibly get here. So it is possible by focusing on like these few key numbers to dramatically increase the output of the field. It will still take us a decade before this starts paying off. Maybe 2 if we have a slow time of getting started on making these things happen. But that's not bad. Let's get started now and make that happen. And then, yeah, open to everyone here. Like, I think if you take this framework, then you should start thinking like, what else could we do to address those key bottlenecks? And maybe there's something missing from the framework. Cool. So that's basically where we're at. And I think I'll end there and take questions.
Wonderful, Martin. Thanks a lot. That was super educational, at least for me. I did enjoy it. So like as a organizing person. So how much time do you have? There are 7 minutes to.
Yeah, I have 7 minutes and then I go.
Okay, guys.
I'll put this thing further behind the meantime.
Yeah, I will not usurp the mic.
You can read more about various things here, so don't ask me about this. Just go read it and then email us later or something.
Yeah, I did share it in various channels of the hackathon, so you can find it there as well.
Cool.
Guys, please raise your hand if you have any questions. We only have 6 minutes. Or write the question in chat as an alternative. While we wait for question. Oh, Tim. Let me see if I need to unmute you. Yeah.
Yeah.
Please go.
Can you hear me?
Yes.
Mm-hmm.
Yeah, nice. So yeah, sorry to raise such a controversial topic. So I'm completely agree with the idea that we need to speed up like generating ideas and stuff, or validating it. Like I remember in space, like Elon Musk's approach when you launch a lot of rockets, then these rockets constantly fail. Like this approach like has shown some effectiveness. And if we go to our area of biotech research, yeah, probably it's off top, etc., etc. So to validate our clocks or like in general to test some ideas, what do you think about like testing drugs on some like activists, humans with their consent, skipping some like stages of the clinical trials? Like we take some risk and we speed up the development like quite significantly. So is it an absurd or not?
I mean, the biggest. So a lot of this comes into like your utilitarian function, right? Like for society, should we be more open to negative events in clinical trials because the status quo is already pretty negative for a lot of people, right? And I think there's a recent kerfuffle around Sarepta, and they had a gene therapy, and like people keep dying on this gene therapy. I think they're up to 7 now. So that is clearly bad. People dying is not good. At the same time, this disease is like lethal. And so everyone who's not getting the therapy is also dying like a couple of years later. It seems to me like we are approaching this from like we as in society, FDA is kind of like, well, do no active harm, right? Like they would let the trolley, drive over the 10 people and not redirect the trolley to the one person, because of the way that, like, very fairly, what gets into the news and what gets blamed and so forth, right? And so from that perspective, if you were purely trying to like optimize the number of healthy years for the US population, you would do more risky trials. You do challenge
And
trials.
my idea is that probably like countries like China, or like you mentioned China, what already doing this, like something like this we don't know, and that's probably one of the reasons we will like be not the first. Yeah, thank you. Thank you.
And so I think if you're like, cool, maybe I'm gonna go do that in Dubai or whatever, right?
Yeah, in some African countries.
Yeah, exactly. So you could go do that somewhere else. I mean, you could do that in the US as well. The key thing is to do it robustly, right? And like get people who design clinical trials involved. I think that if a bunch of activists wanted to go generate human data and just be like, look, this is not we're doing our own clinical trial or something, the key risk is that they do it poorly and like it doesn't do anything because it's like heavily confounded in some way, right? Like, and so doing that correctly possibly could have some like benefits. The tricky thing for humans, right, of course, is like, well, what is the kind of trial? We have this catch-22 where we don't know what the appropriate rapid measurements for aging are, and we don't know what the human anti-aging drug is, right? We don't actually know whether rapamycin works. It has maybe good odds because it works in mice, monkeys, etc. But maybe it'll have a 2% effect size in humans, and so maybe your trial is going to be super underpowered. If you don't actually measure the outcome, it's hard to say what you're doing.
Yeah, we also might end up like a stem cell market where like a lot of stem cells been applied, but we don't know about the efficacy. Mark?
Yeah, exactly, right? So you get this whole sort of like not very serious longevity stuff that as long as it's just circular reasoning, right? Like the clock works because we know that partial reprogramming is an aging therapy and partial reprogramming makes the clock go down. So we know it works, right? This kind of circular reasoning, we have to like make sure that we don't do. That is why the clock assessment program that I'm proposing starts in mice. Clearly humans would be better, but just start with something that you can actually say, this is robust. I am, not conjecturing about outcomes. I'm measuring the outcomes I'm trying to predict.
Mark?
Yeah. I had I'm not sure I understand you have time constraint, but I want to go through briefly the whole pipe and then maybe have a question on it. So you say we're basically generating just a few ideas, and then basically we're testing all of the ideas, and then we also have the last part of making industrial scale of solution of some drug. And then your solution is to say that we need to do biomarkers to test ideas faster and not wait, 10+ years to find out if the idea is good or not. But if you're telling me that we're generating 5 ideas every year and we're testing each one of them, isn't then the problem that we need to test more ideas, to generate more ideas and not to test them faster? I mean, I understand the feedback.
Yeah, no, I think that's right. There's not one thing that is like sufficient to fix the field. And so the way I think about it is sort of like a cascading set of bottlenecks. The current bottleneck is the number of ideas that we have.
Yes. And so if we speak of
so we need to make this number.
Yeah, the 2 to 5 number, which is pretty
We
sad.
need to make this number go way up.
Yeah.
If we do that, then we will run into like how many of the ideas can we actually test as the bottleneck. Because this one will take longer to generate data, we should assume that we get there. This is sometimes if you're playing, Magic the Gathering or poker or whatever you play, right? Or any game. If you're default going to lose, sometimes you have to make the play that is the only play that can potentially make you win. And I think to me that is, yes, we must increase this and we must get ready for this number to be high, even though it is not yet.
Yeah. No, I understand. Like you have to just work everywhere, right? But if the bottleneck is generating enough ideas, is the issue that we have? I mean, I understand the issue is everywhere, but is it that we have too little people, and thus those people are not generating good enough ideas, or is it or as in too little people and we just need more people? Or is it the people we have now are just not generating high quality enough ideas because, let's say, scientists have wrong incentive system and they get, I don't know, incentivized to study some protein that doesn't really move the needle? So how do you think about that? Is it just more people, or improving the people we have and giving them better system?
I think it is both. And actually then the ideas thing here is a little bit of a misnomer, right? Because it's not like, hey, I think that maybe if I whatever electroshock myself, or like if I eat this random plant,
Yeah,
like that
yeah.
doesn't count as an idea, right?
Yeah, yeah,
It's
yeah.
not a therapeutic idea until you have some data. So you have to generate some data, right? So generating data is slow. We have 40 people at Gordian, right? Like because we just like have to physically move a bunch of samples around and do stuff. So I think that's one constraint. There are just like not enough people in the field overall. I also think that the people are not, I mean, it is absolutely true that the people in the field are not consistently taking the most impactful actions, because we have so many, like you can look at caloric restriction. How much could it possibly give us in terms of lifespan, right? And like it depends on like what you say, but like let's even say 10%. I think that's a very high estimate. If it could give us 10%, but like freaking like 15% of the field is working on it, like why are we doing that? Right? I think maybe partial reprogramming has like a good balance, and like maybe even, enough or too much or whatever, right? But so I do think that like there are many people working on something that, if our goal is, and this is not everyone's goal, but if our goal is an output production of healthy years, then they are not doing optimal activity. And when I was in academia, I was not doing optimal activities, right? That's part of
Yeah.
my
It's
proof
rarely,
point.
sadly.
So I
Yeah.
do think that there's a thing there. And there are some labs that are like going a bit wilder, like Vadim Gladyshev, right? Where they are like, oh, let's test things that could be, quite different.
Yeah.
You have to balance that with being able to pull stuff off, of course.
And can I mention just last thing? You talked about the faith, and that's the issue. I think it's maybe most people don't actually perceive aging to be a problem. So I mean, there are some who don't believe we'll do that, but most people just don't think we should solve it, right? So I think that's a crucial part of the, getting more funding and getting more people.
Yeah, I think that's true. Fable of the Dragon Tyrant, yes, still applies. I think so. I kind of like, some of our investors describe what they call the Mr. Burns test. They invest in a lot of, climate solutions, for example, but they tend to invest only in things that Mr. Burns, from The Simpsons, the like evil capitalist guy with a nuclear plant. So only if Mr. Burns would invest do they want to invest. They don't want to invest in things that are like basically half philanthropy, impact investing, or whatever, right? Because those things are going to be so much harder. You're never going to get tailwind. You're never going to get like all of capitalism just like backing you up, right? And so I think for that's where, to me, yes, people considering aging a disease, moral imperative, those kinds of things are legit. And I'm glad that there are people working on like shifting that mindset, for example. That was part of how we started investing a lot in cancer, right? Like Mary Lasker and so forth, like getting Nixon to declare war on cancer. At the same time, it's just so easy if people are going to make money from it. Right? And you see that from the whole longevitainment industry, where, selling whatever will make people money. There's so many people doing it. I bet there are more people doing that than like doing aging science, right? So I think in general, if we can just like align ourselves into, like where the capitalist forces are reinforcing us, that's a good strategy.
Yeah.
Okay, yeah, I think we need to let Martin go, but we can stay here and continue discussing it when Martin leaves. So don't go
Cool.
anywhere. Martin.
Yeah, you can always put the follow-up questions on social media and see if maybe somebody else has a better answer than me.
Yeah, Martin, thanks a lot. It was not
All right,
easy to
thanks a lot
get.
for working on this.